Hello everybody. I'd like to start by thanking the organisers for giving me this opportunity to talk today. I'm going to be talking about our lab's work in understanding and hopefully abolishing growth immunity trade-offs or growth immunity antagonism in Arabidopsis roots, and hopefully in the future crop plants. So just a very quick reminder, we've heard a bit about pattern-triggered immunity already, but pattern-triggered immunity is based on the detection of conserved molecular patterns, such as Cy22 from bacteria, by plasma membrane-bound receptors, and this induces a number of defense responses or immune responses. Firstly, we see, of course, calcium ion influx and ROS burst. We see a MAP kinase cascade, and we also see immunity gene induction, leading to defense responses such as antimicrobial compounds and so on. So these three responses, the ROS burst, MAP kinase cascade, and immunity gene induction, are what I'm going to be referring to as immunity throughout this talk. Okay. So we can induce these immune responses in Arabidopsis roots in the lab in the absence of any pathogens. And you can see that we can detect the ROS burst here, MAP kinase phosphorylation, and immunity gene upregulation in Arabidopsis roots after FLAG22 treatment. However, we also, as we've heard, see an accompanying inhibition of root growth. So we can see this both in Arabidopsis in the lab. We see shorter roots after FLAG22 treatment, and a slowing of root growth in a number of Brassica species. So this is the well-known phenomenon of growth immunity antagonism. And this is important because the losses due to immunity are on a similar magnitude to losses due to pathogen attack itself. So field trials with barley 20-odd years ago showed that after infection with non-pathogenic powdery mildew, so this is powdery mildew that cannot cause disease in the barley, there's a 10% yield loss. And in the lab, if we have constitutive immunity in Arabidopsis roots, in Arabidopsis plants, there's a very significant reduction in biomass. So it's clearly of biological significance. And so our question is, how does immunity inhibit growth? Now, the sort of accepted current explanation for this is that it's simply a case of resource allocation, i.e. that when immunity is off, metabolic resources can be dedicated towards growth, but when immunity is triggered, then those metabolic resources have to be rediverted to defense and therefore aren't available for growth. However, the fact that we still see this trade-off in nutrient-replete conditions and the fact that it can be somewhat uncoupled in the presence of certain microbes suggests that there's some sort of alternative explanation or additional explanation, and there's increasing evidence for a signaling conflict. And so I'm going to be presenting the evidence we have for part of this second explanation. Okay. So let's look first at what growth effects does immunity actually have on the root. So we had a look at the root structural architecture after FLAG22 treatment, and we see that the general effect is one of root shortening. So the primary roots are shorter, the lateral roots are shorter, but there's no growth restructuring of the root system. The number of laterals is the same, but because it's on a shorter root, we see a higher density of lateral roots. Okay. So that led us to question, okay, so what aspect of growth is actually being affected? So we looked first of all, of course, at the meristem, which is organized like this. So we have the quiescent center, which is 2 or 3 cells that act as the organizing center of the meristem. These are very slowly dividing cells, and these organize the stem cell niche, which are the immediately surrounding cells, which are the initials that give rise to the cell files above the root tip, and we also see the columella root cap cells, which differentiate without multiple rounds of division. So our first question was, does immunity disrupt stem cell patterning and disrupt the quiescent center and stem cell niche? And the short answer is essentially no. So we looked first at the number of quiescent center cells, and we see that after FLAG22 treatment, the average of 2 or 3 quiescent center cells is retained. WOX5 expression, which is the transcription factor that's specific to the quiescent center, which is crucial to much of its functioning, also not significantly changed. And we can also stain the columella root cap layers using Lugol staining, which stains the starch bodies. And we see that the number of cell layers in the root cap are also retained. So the immediate root tip meristem is intact. We also see that the patterning of the cell types is not significantly changed. So we've got a number of cell line specific markers here, and you can see that after FLAG22 treatment, which is on the right of each of these pictures, that the patterning of the cell files is not changed. So the next question is what actually is being affected. So the quiescent center and stem cell niche are intact. We just essentially worked up the root. We next looked at cell proliferation. So the reason we wanted to look at this is that when we studied the root structural architecture changes, we noticed that the first emerging lateral root after FLAG22 treatment appears nearer to the root tip. And as lateral roots can only emerge from mature differentiated tissue, this suggests that the differentiation is occurring nearer to the root tip, i.e. that the meristem may be shortened. So we studied this directly, and we do indeed see that after FLAG22 treatment, there is a reduced length of meristem, and if we count the meristematic cortex cells, we see that's a reduced number of cells in the meristem. This led us to question whether cell division is being impaired, and we can use the marker for cell division, cyc B11, and we see that the length of the division zone is decreased, as is the number of dividing cells. So this strongly implicates an inhibition of division in the meristem as being the link essentially between immunity and growth inhibition. So what determines cell division? Well, this is reliant on the mitotic cell cycle. So let's have a quick recap of the mitotic cell cycle in plants. Not as well studied as in animals for obvious reasons, but it's carefully regulated and the general principles are the same. So we have 2 gap phases, separating the synthesis or S phase and the mitosis or M phase, and the transition from the gap phases, so the G1 to S transition and the G2 to M transition, are closely regulated they're positively regulated by cyclin-dependent kinases in complex with cyclins, and these inhibitors of cell cycle genes. So this is in the active state of the cell cycle. If we have an inactive or inhibited cell cycle, you see that the RBR1, KRPs, SIM and SIMR proteins are key inhibitors of the cell cycle. Now, our hypothesis is that immunity might shift the cell cycle in the meristem from this active to this more inactive phase. So some evidence we have of this is that we see a reduced amount of phosphorylated, that is inactive, RBR1 after FLAG22 treatment. So you can see here we have antibody against phosphorylated RBR1, and you see that the amount of it is reduced, suggesting there may be more active RBR1 that may be inactivating the cell cycle. So our next question was, if the cell cycle is key to this link between immunity and growth, could we uncouple immunity from growth by overstimulating or supporting the cell cycle? So what we have done is screened a number of cell cycle regulator mutants and overexpressors to look for an aberrant growth response to FLAG22. And we have found that overexpressing CDKB11 supports growth under immunity. So we see that so here you see in wild-type COL0, if you have increasing concentrations of FLAG22, you see quite a sharp decrease in the primary root length. But in the CDKB11 overexpressors, this root length is supported at all but the highest FLAG22 treatment. So the growth is maintained under immunity. And we see a similar effect on the meristem length itself. So we're able to sort of retain that root length. Crucially, this is without compromising immunity. So in the CDKB11 overexpresses, we see the same immunity gene upregulation. So FRK1, which is a key marker of immunity, is upregulated in both COL0 and in CDKB11 after FLAG22 treatment. We still see intact MAP kinase cascade, or MAP kinase phosphorylation, as you can see here. This is the CDKB11 overexpresses, this is the COL0. And we also see that the FLAG22-induced resistance to Pseudomonas syringae is also intact in the overexpressing lines. So if you treat wild-type plants with FLAG22 and then infect with Pseudomonas, they are less infected than without being pre-treated with FLAG22, and we see that same effect in CDKB11. So in conclusion, just to summarize what we found, we can induce immunity with FLAG22, but this leads to growth inhibition. We've looked at what this growth inhibition actually, what form it takes, and we see that the meristem is intact, the cell types are intact, but the division zone is decreased, and that's led us to the cell cycle. We see some evidence of cell cycle inhibition, and we can support continued growth under immunity by overexpressing a key cell cycle regulator, CDKB11, without impairing immunity. So this opens up a number of other questions and avenues to explore, obviously at the early stages of this, and have a lot of new questions to answer. One of the things this tells us is that CDKs maybe are limiting to growth. It answers some of the how of immunity growth inhibition. We haven't yet got to the why, or the why there might be an active signaling for immunity to inhibit growth rather than it being an incidental trade-off. There's lots of questions. But yes, I wanted to present that to you today. Thank you. Thank you very much.